Okay, welcome everyone to another week of the Beck Speaker Series. We meet every week of the academic year on Mondays at this time, and in this room where we are starting to meet more regularly now with some in-person talks lined up. So that's really exciting for everybody to be back here. You can find the full lineup of speakers for the rest of the year on the webpage at beck.ucla.edu. I'm just going to give you a quick preview of next week's talk, and then I'll introduce today's speaker. Next week we have Tina Lasisi coming, and her talk is entitled Hair, Skin, and the Racialization of Human Variation. That will be in person here, so please join us. And then this week, from our very own departments of computer science, human genetics, and computational medicine at UCLA, Sriram Sankararaman is here, and he's going to be giving us a talk on the role of archaic admixture in human evolution. So welcome.
Great. Thank you. Thank you for the invitation. It's great to be here in person. Am I audible over the Zoom? Okay, great. All right. Today I'd like to tell you some work that we've been doing in my lab over the last few years, trying to understand what we call archaic admixture and how it has played an important role in human evolution. More broadly, in my lab, we study a few different questions, all centered around understanding genomic data. So one set of questions are focused on how genomic data plays a role in modulating and shaping what we call complex traits. So these are traits that are modulated by many different genetic variants together with environmental factors. Another important role that we are trying to understand is how genomic information, genomic data can help us improve treatment, improve clinical outcomes. A lot of this is in collaboration with the UCLA hospital across from us. And today I'm going to focus on the other question, the third, which is how we can learn about evolutionary processes from genomic data. And underlying all of this, we also develop statistical tools, tools from fields like machine learning that help us interpret and analyze genomic data. So I'll give you a little bit of a peek into the types of tools, won't get into too many details, but of course, happy to chat offline or after the talk. And for those of you who might be interested in following up, so my office is in this Engineering 6 building, which is across from the Luskin Center, so you can feel free to come by and chat anytime. All right, so the basic outline for today's talk is to give you a little bit of an overview of this process of what we call interbreeding, admixture, or integration between present-day humans, so ancestors of all people living today, and these archaic humans, which don't exist, but we know of from the fossil record. So I'll first talk about introgression from Neanderthals into modern humans. Then I'll talk about introgression from the Denisovans, another archaic group. And then more recently, we are beginning to realize that it's not just Neanderthals and Denisovans for which we have genome sequences available. There are unsequenced genomes that have also introgressed into modern humans. We call these ghost archaic populations. And finally, I'll talk about understanding the impact of these introgressed bits of DNA in terms of how they've shaped human biology. So this is about the phenotypic impact of Neanderthal DNA. And at any point, feel free to interrupt and ask questions. This can be completely informal. So genetic analyses over the last couple of decades have given us a fairly good idea of the history of present-day humans. For example, we know that modern humans evolved in Africa around 200,000 years ago. And then there was this migration out of Africa, what we call the out of Africa event, the out of Africa bottleneck, that led to the founding of populations outside of Africa today. Now, at about the time when this migration event happened, we had another human population, the Neanderthals, that existed across large parts of Europe and Central Asia. So based on the fossil record, we know that Neanderthals appear in the fossil record at around 300,000 years, and then they disappear about 30,000 years. And if you look at their geographic spread, you can find them in terms of the fossils existing all the way from Europe to places in Central Asia. So modern humans and Neanderthals overlapped both in space and time. And so a question that arises based on the patterns of migration and the existence of these different populations was that whether there was contact and whether this contact led to interbreeding or exchange of genes between these different populations. So the first approach to look at this problem from genetics required genome sequences from the Neanderthals. So the first genome sequence, which we call the draft genome sequence, was obtained from 3 individuals who were found in this cave in Croatia. And so this was a fairly noisy genome sequence, but nevertheless it allowed us to ask this basic question. If we took the Neanderthal genome and we compared it to an African genome, so genome of an African individual, as well as genome of any individual outside of Africa today, we could ask, is the Neanderthal genome genetically more similar to one of these genomes than the other? And the key finding based on these kinds of comparisons is that Neanderthals tend to be genetically closer to non-Africans than to Africans. So this was a surprising finding at that time, and one possibility that it hinted at was that there was interbreeding between the ancestors of all present-day non-Africans as they left Africa and the Neanderthals. So because of this interbreeding, we would expect this pattern of Neanderthals being closer genetically to non-Africans than to Africans. However, this was not the only possible hypothesis that was consistent with the data. There was yet another hypothesis, and this has to do with the fact that there is structure in the African populations. So we call this the ancient structure hypothesis, in contrast to the admixture hypothesis. So the ancient structure hypothesis goes as follows. You have essentially different populations that have existed in Africa that are not completely well mixed. So these are populations that are essentially genetically quite distinct, although they might have low levels of gene flow. And one of these ancient populations then migrates and becomes the ancestor of present-day Neanderthals. And one of the populations that was closer to Neanderthals historically then migrates to become the ancestor of present-day non-Africans. This is a completely plausible hypothesis given what we know about the diversity of the fossil record in Africa. However, they make distinct predictions. So the admixture hypothesis essentially says there must have been contact, there must have been exchange of genes between modern humans and Neanderthals fairly recently. So it must have happened after the split of Africans and non-Africans. So this is an event that must have happened within the last 100,000 years ago, depending on when we think the out of Africa migration happened. On the other hand, the ancient structure hypothesis predicts that the last exchange of genes between modern humans and Neanderthals must have been quite old. It must have in fact predated when modern humans appear in the fossil record, so it must have been at least 230,000 years ago. So now the question is, could we try to differentiate these hypotheses given the genetic data that was available to us? So to do this, can we try to time or date when gene exchange happened last between modern humans and Neanderthals? So to be able to date a potentially old admixture event between archaic and modern humans, we use this property of our genomes that get transmitted. And this has to do with the fact that every time our genome gets transmitted from parent to offspring, there is this process of shuffling that happens, a process called recombination. And as a result of this, recombination happens at a predictable rate. We can use recombination as a clock to be able to time these ancient admixture events. So the broad idea is something like this. So imagine that this is a modern human genome, and this is an archaic human genome. And these two individuals come together and interbreed. So in the first generation, an individual like this is an offspring of the modern and the archaic humans. So this individual inherits a full chromosome from their modern human parent, and another full chromosome from their archaic human parent. Now, when this individual produces offspring, if you look at their genomes, these offsprings now begin to have parts of their genome that are modern human interspersed with parts that are archaic. So this is because of recombination, so it ends up essentially shuffling up which parts of the genome get transmitted from parent to offspring. But if you look at the parts, essentially there are large chunks of genomes that are entirely either modern human or archaic in ancestry. So that's because recombination hasn't had enough time to shuffle up these segments. But now if you let this process run, if you look at what happens, say, thousands of generations into the future, or what happened today given an event that happened thousands of generations in the past, the genomes will look like this, where you have this mosaic structure where there are regions of archaic ancestry interspersed with regions of modern human ancestry. So what this process gives us is an ability to time these admixture events by inspecting the lengths of these archaic human segments. So this was something that we then computed. So we devised a statistical test which would look at not necessarily the lengths. Turns out getting the exact lengths is a hard problem. We'll talk about it in just a bit. But it looks at the correlation between different positions that were inherited from these archaic humans across distance. And the key idea is the correlation decays with distance because of this recombination process. And how fast it decays gives us a measure of time. So now we can measure the rate of decay of this correlation with distance. And when we do this, we get an estimate of the date of interbreeding, which is between 47 to 64,000 years before present. So this suggests that interbreeding occurred fairly recently, and in fact it lends support to the fact that there indeed was interbreeding between modern and archaic humans. So since this method, this analysis was published, there have been other lines of evidence that have also got at this in a more direct manner. So what has happened since we published our study is there have been future studies since then which have looked at the genomes of what we call ancient modern humans. So these are people who were living thousands of years before present, tens of thousands of years before present. And these are individuals who lived in outside of Africa. So if this prediction is correct, you could now look at these individuals, and they must have larger regions that have been inherited through this interbreeding event because they lived, say, 10,000 years before present. And so now you can back out dates from those genomes, and you get essentially dates of interbreeding which are consistent with the estimates that we obtained. So this is about the time of interbreeding. We can also estimate how much interbreeding there was between Neanderthals and present-day humans. So what fraction of the ancestry, of the genetic ancestry of present-day non-Africans traces to this interbreeding event. And again, there are some statistical nuances to this, but overall, all of these approaches, they end up with this estimate of about 2% of the genetic ancestry of present-day non-Africans tracing to this interbreeding event. So the big picture that comes out of these analyses is one where we have a split between Neanderthals and the ancestors of present-day humans. Then we have the split between ancestors of present-day non-Africans and Africans. And then we have this interbreeding event, which introduced Neanderthal DNA into the ancestors of present-day non-Africans, followed by the diversification of these populations into present-day populations outside of Africa. So this is a kind of a global picture of how Neanderthal introgression, Neanderthal interbreeding has shaped human evolution. So a question that arose at that time was, is there any kind of biological impact of this Neanderthal DNA? So as you see from this previous picture, so Neanderthals were separated from modern humans for quite a bit of time, a couple of hundreds of thousands of years, depending on different estimates. So essentially, there were mutations that arose on this Neanderthal lineage that were never present in any of the modern humans. So this interbreeding event was essentially an event where a large number of novel mutations were introduced into the human populations. So you could hypothesize that an event which introduces large numbers of novel mutations in a relatively short span of time could have had a biological consequence, either positive or negative. So the question is, can we try to understand the consequences of these mutations that were introduced by this interbreeding event? So one of the first attempts at this happened through a study that was looking at a particular disease. In this case, this was a study that was trying to identify genetic risk factors for type 2 diabetes. So this was a study that was done in individuals of Mexican American ancestry, and the study came up with a new discovery, a new genetic variant associated with increased risk for type 2 diabetes in Mexican American populations. Now, when we looked at this variant and asked, can we try to understand how this variant, this risk variant for type 2 diabetes is distributed in populations across the world, we find a pattern that looks like this. So what we are showing here is different populations around the world. The orange part of this pie is basically telling us what is the frequency of the derived allele, which also happens to be the allele that increases risk for type 2 diabetes. So what we see here is essentially the derived allele is absent in Africans. It is present at relatively low frequencies in populations in Europe, slightly higher frequencies in East Asian populations, and substantially higher frequencies in populations in America. So this is the pattern that we might expect if this was a variant that was introduced into human populations through interbreeding, because interbreeding happened after the split of African and non-African populations. And then when we look at this risk variant and we compare it to the analogous region in the Neanderthals, we find essentially that the risk variant matches the variant that is found in the Neanderthal genome. So this is likely a variant that was introduced into modern human populations through Neanderthal introgression, and this is something that has been confirmed by subsequent modeling analyses. So this was one of the early examples of Neanderthal introgression having shaped or contributed to specific phenotypes. And in fact, there have been other notable examples. So there have been studies from other groups that have shown significant contribution of Neanderthal introgression at other types of genes. So STAT2 and HLA are genes that are especially important for immune-related functions. And so this is notable because this tends to be consistent with the hypotheses that Neanderthals, they were in these environments where they had been adapted for hundreds of thousands of years, and these were environments into which modern humans were moving. And so immune-related genes were particularly beneficial for modern humans to acquire through introgression. But these are individual examples. The question is, what can we say more broadly? So apart from these individual examples which are of interest, can we go to a genome-wide assessment of the contribution of Neanderthal introgression? So to do this, we need to build maps of Neanderthal DNA. So essentially what we need to do is go and identify which are the places in our genomes that carry traces of Neanderthal inherited DNA. So going back to this picture, so if you look at individuals today who have descended from this introgression event, their genomes have this mosaic or patchwork of regions which are Neanderthal in ancestry versus regions that are modern human in ancestry. And what we'd like to be able to do is essentially paint the genome according to the ancestries of these individuals. So to do this, we need to build statistical methods. So these ancestry identifications, these colors are not available to us, so this needs to be inferred from genomic data. So the next step was to be able to build maps of Neanderthal DNA. And the key idea behind these approaches, these statistical models, is we look at genome of a test individual. So this could be genomes from any of the present-day human populations. We compare this to an archaic genome, in this case the Neanderthal genome, and to a modern human genome, which presumably doesn't have large amounts of archaic DNA. So in these kinds of examples, we typically use present-day African genomes as representative of genomes which have relatively little archaic DNA. And we are then asking which parts of the genome of this test individual are closer to the archaic relative to the modern human. So that allows us to paint this test genome as inheriting distinct chunks of modern human versus Neanderthal DNA. So here's an example. So there are different pieces of genetic information that go into building these probabilistic models. So here's an example of one piece of genetic information. So here we are comparing a non-African genome to a Neanderthal genome as well as multiple African genomes. And at this position where there is a mutation, the non-African genome carries the allele that matches the Neanderthal, and this allele is absent in all of the Africans. So this was the kind of pattern that we used in our type 2 diabetes genetic risk factor example. And so seeing a pattern that looks like this would increase the probability that this allele was inherited through interbreeding from the Neanderthals. This is just one type of information. In reality, what we do is we combine other pieces of information. For example, we don't just look at a single SNP, we can look at combinations of SNPs. This is what we call haplotypes. And so we can ask, is a haplotype more similar to Neanderthals than to the African genomes? And of course, we have information from the time of admixture. Because we know the admixture happened about 50,000 years ago, there is an expectation of how long these segments need to be. So all of this information goes into predicting or making inferences about these maps. So now we apply this approach to genome sequences from what is called the 1,000 Genomes Project. So here is an example of what the model predicts. So here we have 3 genomes. We have the genome of a European American, an East Asian, and a sub-Saharan African genome. And what we have plotted here is, as you move along each of these genomes, what is the probability that a given position along that genome is Neanderthal in ancestry? So the higher we are towards 1, the more confident we think that that particular position in that individual is inherited from Neanderthals. So the first picture that comes out of this is there is quite a bit of such regions which are confidently predicted to have Neanderthal ancestry in European and East Asian genomes that we have here, and relatively few such regions in the sub-Saharan African genome. So again, consistent with the model of Neanderthal introgression that we have informing us. We can ask overall what proportion of genomes are predicted to be Neanderthal in ancestry across different populations. So in Europeans and East Asians, it's again of the order of 1 to 1.5% across all the individuals that we then could analyze. In the African populations, it was considerably lower, again consistent with our expectations. It's not 2% because again the model has some difficulty in finding Neanderthal DNA at specific regions of the genome, which are complicated, which are varying quite a bit. So it's not pulling all the Neanderthal DNA out that we would like. We can also be more quantitative about this. So we can ask what proportion of these calls of Neanderthal DNA are false positives versus how much of the Neanderthal DNA do we pick out. And what we find is it can actually be quite precise. So our false positive rate is about less than 10%. And with that false positive rate, we can pick up anywhere between 70 to 80% of all the Neanderthal DNA out there. So what I showed you previously was an individual map of Neanderthal ancestry. In other words, for each person, what their Neanderthal ancestry looks like. We can also look at this at the population level. So we can average across all individuals, either in Europe or in East Asia, and build these population maps of Neanderthal ancestry. So here we have one such map. We have the 22 chromosomes along with the X chromosome. And as we move along these chromosomes, what we are plotting here is what is the frequency of Neanderthal ancestry. In Europeans, that's in red. In East Asians, in green. So what comes out of this picture is the fact that there is tremendous variation in Neanderthal ancestry as we move along a person's genome. So we quoted this number of 2%. That's an average. So if you add up all of these numbers across a person's genome, you get a number between 1 to 2%. But clearly, there is quite a bit of variation, substantial variation across the genomes of these populations. So we have these peaks of Neanderthal ancestry. So that's the first thing that we see. For example, there are regions where the frequency of Neanderthal ancestry today is greater than a half. In other words, more than half the individuals present today, say either European of European ancestry, carry the Neanderthal version of a given gene. So for context, about 50,000 years ago, this was about 2%. So there has been an increase in the frequency of Neanderthal ancestry at these regions from about 2% 50,000 years ago to greater than half today. So these regions turn out, and this is something where we have done additional modeling to show, are likely to have been regions that have risen up in frequency not by chance, not by genetic drift, but because they must have had some adaptive impact. So they were adaptively beneficial, which is why they have risen up in frequency in the population that they have introgressed into. So here's an example. This is one of the most extreme regions. So this is a region which has, again, about 60% frequency in present-day Europeans. And it turns out this is a region that overlaps this gene called BNC2. So this is a gene that has been shown to be important for skin-related phenotypes. So it has been shown to be associated with functions like skin color, freckling, hair color, and so on. So there are many possible reasons why this could have risen up in mutation in frequency, but it does seem to have something to do with either skin or hair-related phenotypes. We asked this question more broadly, where we didn't look just at the top region. We asked if we look at all the regions which seem to be elevated in Neanderthal ancestry, are there specific biological processes, biological annotations that seem to be associated with these genes. So we performed what's called a gene ontology or a Go analysis of these regions with elevated Neanderthal ancestry. There are several Go categories or Go biological processes that pop up, but one thing that's quite consistent across many of these analyses is there is an enrichment for skin and hair related phenotypes or keratin biology in these regions. So that was about the peaks, but the other interesting aspect of these maps are the deserts of Neanderthal ancestry. So there are large regions of the genome where essentially there appears to be no Neanderthal ancestry across any of these populations. So in fact, what we are able to identify are multi-megabase long regions of the genome, so at least several million bases long regions, which are devoid of Neanderthal ancestry across all of the populations that we have analyzed. So here's one example of one desert of Neanderthal ancestry. This lies on chromosome 7. And this turns out to be a particularly interesting region because it overlaps a gene called FOXP2. So FOXP2 is an interesting gene in human evolution because this is a gene where there have been mutations that have been found to lead to speech and language disorder. So clearly this is a gene which has been shown to be important for speech and language related phenotypes. So the hypothesis, and this is not something that we know for sure, is that these large deserts of Neanderthal ancestry are regions where there has been selection against the introgressing Neanderthal ancestry. So these regions are resistant to Neanderthal introgression. Of course, these regions are quite large, and this is not the only gene that is present there. So being able to identify what might have driven this selection against Neanderthal ancestry is still very much work in progress. So that was just a qualitative view. We looked at these deserts, but here is a quantitative view of trying to understand what is happening genome-wide to Neanderthal ancestry. So what we then did is we looked at regions of the genome binned according to this statistic, which we call the B statistic. So this is a measure of how functionally constrained different regions of the genome are. So we know that certain regions of the genome tend to be conserved across evolution, presumably because they have functional importance. And within each of these B statistic bins, we can ask how much Neanderthal ancestry do we observe based on our maps. So as we move from left to right, we are moving from regions which are functionally constrained. So there's strong functional constraint on the left, to relatively loose constraint on the right. So regions on the right are more likely to be evolving neutrally compared to regions on the left. And what we see is regions where there is strong constraint also have reduced proportion of Neanderthal ancestry, both in Europeans and in East Asians. So this is consistent with this hypothesis that overall, across the genome, there seems to be selection to remove Neanderthal ancestry, purifying selection that has reduced the amount of Neanderthal ancestry that exists in present-day populations. So now we can ask what might have led to this purifying selection. So one idea, and this is some work that has come up that was done in other groups, is this idea that Neanderthals across the hundreds of thousands of years when they were separated from modern humans had a very low effective population size. So today when we think of populations with low effective population sizes, we think of founder populations like Ashkenazi Jews, populations in Finland. Neanderthals have had effective population sizes which are substantially lower than any modern human group today. As a result of this, they have accumulated a large load of deleterious variation that then entered the modern human population and was selected against. So this has been one hypothesis, and a very likely hypothesis to explain this reduction in Neanderthal ancestry near functionally constrained regions. Another possible hypothesis that could explain this is this notion of male hybrid sterility. So this is this idea that when you have two populations that are diverged substantially, there are genetic mutations that arise on each of these populations that are completely neutral. But then when you come together and put them onto the same background, this pairs of genetic mutations tend to interact and lead to reduced fitness, often to sterility. So a classic example of this is trying to cross a horse and a donkey to form a mule. So the reason why the mule is infertile is because of this kind of a mechanism. So the question is whether this might explain some of this reduction in Neanderthal ancestry that we are seeing. So this hybrid sterility hypothesis, it makes two predictions. The first is that hybrid sterility tends to be particularly strong on the sex chromosomes. So now we can ask what happens to Neanderthal ancestry on the sex chromosome, on the X chromosome, compared to the autosomes. And what we find is Neanderthal ancestry, both in Europeans and in East Asians, is substantially reduced on the X compared to the autosomes. Hybrid sterility makes a second prediction. Generally, hybrid sterility tends to be quite strong in genes which are expressed in testes. So we can then look at the regions of the genome which contain genes expressed in testes and ask what happens to the Neanderthal ancestry in these regions. And we find that Neanderthal ancestry tends to be depleted in genes expressed in testes, again in both populations, consistent with this hypothesis. So now I'll switch to the second archaic introgression. This is from the Denisovans. So this was a discovery that was made when this pinky finger was discovered in a cave in Siberia, the cave in the Altai Mountains, called the Denisova cave. And when this, the DNA from this pinky finger was sequenced, it was found to be from a population that is close to the Neanderthals, but not quite, or quite distinct from the Neanderthals. Because of where they were found, this population was called the Denisovans. And like in the context of the Neanderthals, we can ask whether there was interbreeding between this archaic population and other modern human populations. Whereas the Neanderthals contributed genetic material to the ancestors of present-day non-Africans, it was found that Denisovans contributed archaic material through interbreeding to the ancestors of present-day Oceanians. So present-day Oceanian populations, in addition to the Neanderthal ancestry that they've inherited, have also inherited ancestry from the Denisovans. So now we can try and compare the proportions of Neanderthal ancestry to Denisovan ancestry across populations in the world. So this is a study where we looked at hundreds of populations spread across different countries. And when we look at the proportion of Neanderthal ancestry, we essentially see Neanderthal ancestry available in present in populations outside of Africa. But then when we look at Denisovan ancestry, we see that they are primarily found in these populations in Oceania, but then there is also Denisovan ancestry found in populations in South and Southeast Asia. We can also build maps of both Neanderthal and Denisovan ancestry. So this is a map where we have compared maps of Neanderthal and Denisovan ancestry. So the red is maps of Neanderthal ancestry and the blue is maps of Denisovan ancestry. Like we found in the context of Neanderthals, we find peaks of archaic ancestry both for Denisovans and for Neanderthals. So here's one notable example of introgression from the Denisovans leading to a peak of Denisovan ancestry. So this is at a gene called EPAS1. So this is a gene where this mutation has been found to help Tibetans survive in high altitude environments. We also see deserts, like we found deserts of Neanderthal ancestry, we find deserts of Denisovan ancestry. So if we go back to our FOXP2 locus, we find that it's a desert both for Neanderthal and for Denisovan ancestry. So the picture that's emerging is some of these deserts, some of these regions that are resistant to introgression seem to be a shared feature across these different introgression events. We can again be more quantitative. We can ask how does either Neanderthal or Denisovan ancestry vary across the genome, and we find both Neanderthal and Denisovan ancestry reduces in regions of the genome that are functionally constrained. This is true both on the autosomes and on the X chromosome. Denisovan ancestry, like Neanderthal ancestry, is also reduced on the X chromosome compared to the autosomes, and it also seems to be depleted on genes that are expressed in testes. So again, the picture that comes from these analyses is one of purifying selection on these archaic DNA. So this was all about Neanderthals and Denisovans, where we have access to an archaic genome that was sequenced. However, the picture in Africa has still remained relatively understudied, and the reason for this is it's extremely challenging to get access to archaic genomes given the conditions that exist in Africa. So in terms of ancient DNA, we have relatively few high quality genomes that go back this far back in time to allow us to study archaic populations in Africa. However, there's been approaches that have tried to use the modern genomes that we have and that are growing to be able to say something about archaic ancestry in Africa. So we decided to explore these approaches. So the idea is, can we use the fact that there are many modern human genomes that are being sequenced in Africa to be able to pull out what looks like archaic ancestry even if we don't have an archaic genome? There might be patterns in the genomic data that might be informative of archaic ancestry in these populations. So to study this, we used two complementary approaches. So the first one is something called the site frequency spectrum. So I'll talk a little bit about this. And the second one is the approach that we have been using in the context of Neanderthals and Denisovans, which is to look at these introgressed segments of DNA. So let's talk about the site frequency spectrum. So what is the site frequency spectrum? So the site frequency spectrum is a way of taking genetic variation data and summarizing it. So essentially, let's say we have a population that looks like this. So we are looking at individuals from Africa. Each column is, say, a haploid African genome. We would go to each position where there is a variant, and we would count up how many copies of the variant are present across individuals at that position. So this variant, we have the A allele present in 2 copies. So we would build a histogram which makes counts up how many times we see a variant that's present in 2 copies across these individuals. So the site frequency spectrum is a very well studied object in population genetics. The reason why it's well studied is because it gives us lots of insights about demographic history, and selection. The problem is if we try to build a full site frequency spectrum and use it to model the demography of this population, in some sense it has too much information built into it. So we would have to figure out how population sizes in the African populations changed across time to be able to say something about admixture. So this turns out to be a challenging task, so we use a different object which we call the conditional site frequency spectrum. So the conditional site frequency spectrum is the following object. We are going to look not just at the African population here, but we'll also look at an outgroup, in this case the Neanderthals, and we will look at the chimpanzee, which is going to tell us what's the derived and the ancestral allele. So in the conditional site frequency spectrum, we are going to restrict our attention to only those sites where the Neanderthal genome carries a derived allele, whereas the chimpanzee carries an ancestral allele. Focusing on those sites, we will then build our site frequency spectrum as before. So when we do this, we are going to get a histogram that looks like this. So this histogram tells us there is some number of sites that are present in one copy across this population. There are some number of sites that are present in 2 copies, some number of sites present in 3 copies, and so on. So now we can ask, why is this conditional site frequency spectrum so interesting? So here's a prediction. So this came from a paper by Monty Slatkin and colleagues several years back. So what they showed is, let's say you have 2 populations, in this case Africans and Neanderthals. These 2 populations split and there was no further contact. If you look at the conditional site frequency spectrum in African populations conditioned on the Neanderthals carrying the derived allele, the expectation is the conditional site frequency spectrum looks like this. So it's going to be flat, where you're going to see essentially every allele across different frequencies appear about the same time. So this is a theoretical prediction. Now we can apply this to the data and ask, what do we expect? And importantly, why we again care so much about this conditional site frequency spectrum is this prediction holds no matter what happens to the population sizes within Africa. So population sizes within Africa, which we don't know, they're challenging to model, could vary arbitrarily. There could be bottlenecks, there could be expansions. This property of the conditional site frequency spectrum holds independent of any assumptions that we make about population sizes within Africa. Now let's look at data and see what we expect. So this is the conditional site frequency spectrum, either when we condition based on Neanderthals or Denisovans as an outgroup in the Yoruba population. So this is the West African population. And the key takeaway is it's anything but uniform. So what this is telling us is this model, where Africans split from Neanderthals with no further contact, no further interbreeding, is not supported by the data. We can look at other West African populations. So we can look at the Yoruba and other populations like the Mende, the Gambian population in the 1000 Genomes Project, and Isan, and all of them show this U-shaped conditional site frequency spectrum. So this seems to be an event that is potentially shared by these different populations. Of course, there are bioinformatics artifacts that could explain these kinds of results. There could be errors in our genomes, either in the archaic genome or in the chimpanzee genome. So we asked, is that possible? And it turns out these errors would have to happen at a very high rate to be able to explain these results. There are population genetic explanations that might give rise to this. So there are events like GC bias gene conversion, background selection, back mutations, or recurrent mutations. And again, either the rates needed are too high, and we did additional filtering of the data. So you look at regions which are less likely to be affected by these phenomena, and we did simulations which all convinced us that these kinds of events could not have explained this. So this must be a demographic event that explains our conditional site frequency spectrum. We then asked, are there models of human history that can explain this conditional site frequency spectrum? So here we considered more realistic models that have been built by other groups. So this is a model where people have shown there has been introgression into the Neanderthals from the ancestors of all present-day humans. Of course, this is the Neanderthal introgression into non-Africans, and there's also been evidence for gene flow between present-day Africans and non-Africans. So when we put all of these in, we get a conditional site frequency spectrum that looks like this, still quite a bit different from what we see in the data. So a model that does explain the conditional site frequency spectrum is a model of what we call ghost archaic introgression. In other words, we have a population that diverged prior to the human Neanderthal split, and this population, this unknown population, we also call it a ghost population, then introgressed into the ancestors of present-day Africans. So now when we add this event, we now see that our theoretical conditional site frequency spectrum begins to look like our data. We of course explored other possible models. For example, instead of a ghost, could it have been the Neanderthals themselves? Could the Neanderthals themselves have introgressed into present-day Africans, and could that have led to this kind of a conditional site frequency spectrum? We get something that looks like this, again different from our data. So we also do formal tests, and we find that it doesn't give a good fit compared to a model where there's an actual ghost archaic introgression. We've talked about deep structure within Africa. So based on the fossil record, it's clear that there are these populations that are quite distinct, that have existed in Africa over hundreds of thousands of years, and that they might have exchanged genes. So we call this a deep structure within Africa model. And again, such a model doesn't quite explain our data. So again, compared to a model of a ghost population, it is a model that doesn't fit the data as well. So a model of ghost archaic introgression explains this conditional site frequency spectrum. We then did some additional analysis to figure out the demographic details of this model. So what we then inferred is this population had to have diverged prior to the human Neanderthal split. We think it contributed about 10% of this genetic ancestry to present-day African populations, and it introgressed fairly recently, again about 40,000 years ago. Another interesting aspect is this ghost population seems to have a fairly large effective population size. It's about 20,000 effective population size, which is quite a bit larger than the effective population sizes of present-day populations. So what that seems to suggest is this could actually be multiple populations that have coexisted and that introgressed into the ancestors of present-day Africans. So that was the first line of evidence about this ghost introgression event. The second line of evidence is a statistical method that we developed previously and we discussed. But now we're going to pull out regions of archaic ancestry in this test population without actually requiring an archaic genome. I won't go into too many of the technical details, happy to talk about that later. But the key idea is we essentially use the fact that when you have many modern human genomes, you can group the different chunks, and regions which are archaic in ancestry tend to look quite distinct and group separately from regions that are modern human in ancestry. Statistically, we actually have a machine learning model that computes these types of features from the data and is able to predict whether there is archaic versus modern human ancestry at a given region. Before we apply this to the African setting, we wanted to convince ourselves that the model is sensible. So we applied it to the Neanderthal introgression, but pretending we don't have the Neanderthal genome. So we think of this as a positive control. Essentially, we can now figure out that at a threshold at which we are confident, this model predicts about 2% of the genome as being Neanderthal. It doesn't know it's Neanderthal. It's an archaic ancestry that it's predicting. If we look at these regions that it predicts and ask whether it matches the Neanderthal genome, the regions that are predicted to be archaic end up matching the Neanderthal genome much more than regions that are predicted to be not archaic. It also pulls out this BNC2 region that we talked about previously. So that makes sense that it's pulling out features that we have seen based on a method that has access to the Neanderthal genome. And it also predicts this increase in Neanderthal ancestry as you move away from functionally constrained regions. Of course, there is more noise here because we don't have access to an archaic genome. And I think my laptop is going to shut down soon if I don't plug it in. Great. Okay. Okay, so that was just a test run. Now we apply to the African genomes. So when we applied it to the African genome, we find that about 8% of the ancestry in these in the Yoruba and the Mende, which were the two populations we applied it to, are predicted to come from an archaic population at a false discovery rate of 20%. So now we have these bits of DNA that are predicted to be archaic. We can now go back and check. The model doesn't know where this comes from, so we can ask which populations are likely to have served as ancestral populations for this putative archaic ancestry. So we compared these bits of DNA identified by the model to present-day hunter-gatherer and pygmy populations. So we asked, at these regions called as archaic, are they more similar to genomes from these populations compared to regions labeled not archaic? And in fact, we observed the opposite. So these regions labeled as archaic are more distant to individuals from hunter-gatherer and pygmy populations compared to regions labeled as not archaic. So it's basically telling us that these are unlikely to be the source population for this ancestry. We can also go back to our known archaic genomes, Neanderthals and Denisovans, and the picture is the same. The archaic DNA that is identified is not particularly closely related to either the Neanderthals or to the Denisovans. So this is again consistent with the fact that this came from a ghost archaic population which diverged prior to the human Neanderthal split. So this is exactly the pattern that we would expect under this model. Okay, so in the remaining few minutes, I'll now go back to this question of understanding the biological impact or the phenotypic impact of Neanderthal DNA. So we have about 2% of Neanderthal DNA in the genomes of present-day non-African populations. We talked a little bit about the phenotypic impact. We said it's deleterious in general, and this is evidenced by the fact that it's been purged from regions of the genome that are functionally important. But of course, there are individual loci where it's adaptively beneficial, as seen from the fact that it has increased in frequency. But what is still missing is being able to figure out were there specific traits that were substantially impacted by Neanderthal DNA, and whether we can identify how Neanderthal DNA has shaped particular phenotypes. So to do this, we take advantage of the fact that there are now available these large biobank scale genomic data sets. So these are genomic data sets where we have genotype information across hundreds of thousands of individuals paired with very diverse collections of phenotypes. So an important example of this, an example that is widely used, is something called the UK Biobank. So this has genetic information from about 500,000 individuals in the UK with thousands of phenotypes that have been ascertained. The question is, why do we need these large data sets? And this has to do with the fact that Neanderthal introgression introduced about 2% Neanderthal ancestry into modern humans. So what that means is, on average, a mutation that was introduced by Neanderthal introgression is going to be present in about 2% of present-day individuals. So to have statistical power to identify the impact of these mutations, we need these large sample sizes. So to be able to avoid false positives, to have robust associations, we need these large sample sizes. So the way we approach this problem is we went back to our maps. So these are our maps of Neanderthal DNA that we had constructed. And in these maps, we have these mutations that are essentially sitting on these introgressed Neanderthal haplotypes. For example, this A allele here, we identify as a mutation that was introduced by Neanderthal introgression because it's absent in Africans and it's only present in the Neanderthal genome. So these mutations that tag Neanderthal ancestry, we call these NIMs, or Neanderthal informative mutations. So it turned out when this UK Biobank study was being designed, they were designing a genotyping array which would essentially tag most of the interesting variation in the genome, and they were looking for content to add to this genotyping array. And we were lucky enough that at that point they got in touch with us and asked, are there interesting sets of mutations that would be worth adding to this genotyping array? And we at that point had just built this map. So we said it would be interesting for us to be able to study Neanderthal introgression in this large scale study, which would be unprecedented at that time. So we were lucky that the array that was used in the UK Biobank now has this set of mutations which are informative of Neanderthal ancestry that allow us to study the impact of these mutations in these large cohorts across many different types of interesting phenotypes. So now, given this study, we can now ask questions like, do these NIMs, these mutations that tag Neanderthal ancestry, do they contribute to variation in complex traits? So the answer turns out that, like most genetic variants, for most complex traits, each genetic variant has a very small effect on a trait. But in aggregate, you can ask, is there an effect? So this is a quantity which we call heritability. So the heritability of a trait is simply what proportion of the variance in that trait is explained by genetics. So now we can ask how much of that heritability comes from these NIMs that we have identified. So we call this the NIM heritability. So here we have looked at hundreds of traits grouped according to their category. So we have anthropometric traits, autoimmune disorders, blood pressure, eye, kidney, liver, and lung, and skin and hair related traits. What we find is there is a substantial contribution of NIM heritability to anthropometric traits, blood biochemistry, blood pressure, and so on, but not so much skin and hair, which is a surprise because some of these prior studies seem to suggest that that is where Neanderthal introgressed variants might have had a big impact. But on the whole, globally, they don't seem to have had such a huge impact. We can then ask, do these NIMs contribute more to the heritability of these traits than modern human SNPs? So there were SNPs that were never introgressed, then there are SNPs that are introgressed. What happens when we compare their contributions? So this is what we see. What we find is across all the traits, NIMs tend to contribute less to heritability compared to modern human SNPs. So they tend to be depleted in their contribution to different human traits. Some classes of traits more than others. For example, again, anthropometric traits, blood pressure traits, lipid traits, lung-related traits, those tend to be places where there is a substantial depletion of Neanderthal introgressed mutations. This is exactly what we would expect if there is purifying selection against these introgressed Neanderthal alleles. As we said previously, in general, these introgressed Neanderthal alleles have been selected against, so they tend to be away from functionally important regions. We can also account for this. So we can ask, is this depletion something unique about the introgressed alleles, or is it something about the fact that they are in regions which tend to be less functional? And when we account for this, it turns out it's actual selection against the alleles themselves and not just against the functional changes in general. So with these kinds of studies, the big picture that's emerging is there is contributions from these introgressed alleles. They are modulating complex traits. But in general, because of the effect of purifying selection, they tend to be depleted in their contribution in terms of heritability to these complex traits. Okay, so this is very much ongoing work. So some of the questions that we are now thinking about in my lab is we have now looked at populations in Africa and populations outside of Africa. We have found that there is introgression, not just from known genomes, but also unknown genomes. We call these ghost introgression events. We'd like to understand how pervasive the signal is across other populations. So clearly, we have only sampled a small fraction of the genetic diversity of present-day humans. So how pervasive is this signal? How likely are we to find newer signals of introgression? And finally, what is the impact of these events on biology? So we are now getting these types of large-scale genomic data sets not just in populations of European descent, but in other populations. And so this is going to allow us to, for example, understand the impact of the Denisovan introgression event on biology and compare it to what we saw in the context of the Neanderthals. The other key lesson that comes out of this is this idea that introgression is pervasive. No matter what timescales we look at, what sets of populations we look at, we always find these arrows that complicate our understanding of human history. So essentially, we are dealing not with a tree-like structure, as basically Darwin sketched in his book, but this tangled web. And the challenge for us is, how do we actually make sense of this? How do we go from genetic data, which comes out of this process, to figuring out what this underlying web looks like? Turns out that's a very challenging problem. We are only now beginning to understand the difficulty and our ability to say something about these types of events. So that's, again, ongoing work in my lab. With this, I'd like to thank my students who led a lot of the work. So Arun, April, Chris, and Erin were some of the key people who led the work. I'd like to thank funding agencies for the support, and I'd be happy to take questions.
Questions? Brian?
Hi, thanks for your talk. It's great to meet you. Thanks for coming and joining us over here. I was wondering if you could go back and talk a bit about the part of your findings where you were identifying areas of the genome that are resistant to Neanderthal ancestry. You mentioned in specifically the FOXP2 region. I think you also made that point with Denisovan introgression. The context for my question is that my recollection is that the Neanderthals had a similar variant in that region, as do living Homo sapiens today. So is it possible that, well, A, is that true? Or B, is it possible that it's not necessarily resistant, it's just that there was no variant coming from Neanderthals that potentially could have introgressed, because that they shared the same variant?
Right. So yeah, so I guess this is the finding, which is talking about this resistance to introgression. So this is chromosome 7. This is the region that overlaps FOXP2. It's about 10 megabases long. So and this is a place where we find almost no traces of either Neanderthal or Denisovan ancestry. So the first question is about the mutations in the FOXP2 gene and whether those are shared between Neanderthals and modern humans. So the answer is there are these amino acid changes, these changes that change the protein that is being encoded by this gene. And indeed, Neanderthals and Denisovans share the human version of this protein at the FOXP2 gene. However, there are other changes upstream of these genes, so changes in that change the regulation of this gene, and there are differences there. So how much of this is driven by protein coding versus regulation is unclear at this time. The second part is whether these deserts could simply be reflective of the fact that there wasn't introgression of any of these variants. So one thing that we did in our analyses is we asked, are there things about these regions, FOXP2 in particular, but other regions, that would make us hard to detect introgression in the first place? For example, are they unusually devoid of genetic variation? So then we would, if there is no genetic variation, we are not powered to be able to detect introgression. The answer turns out not particularly. So these regions are typical FOXP2 and these other deserts that we have identified. And so that led us to say that this is not just a power issue where we are unable to find introgression that might have existed. The other thing which also makes it striking is not just the fact that it's devoid of ancestry, it's devoid of ancestry over such a large region. So 10 megabases is a large region of the genome. There's FOXP2, but there are many other genes here. And the fact that there is a lack of introgression over such a large span of the genome, that makes it very confident that we are not missing something. So if it was a small region of the genome, just statistically we might be able to miss whether there was introgression here. But when you have a region as large as this, we are able to show that it's extremely unlikely that we are just able to miss out on potential introgression. So these are very likely places where there has been removal of introgressed deserts, introgressed ancestry. Now the question is, because these regions are large, it's actually harder to figure out what is going on there. So there's FOXP2, but there are many other genes, and there are variants that differentiate humans and Neanderthals within these deserts. So what makes them easy to identify as deserts also makes it hard to pinpoint what are the changes in these deserts that are functional, that could have led to them being deserts.
Thanks for your talk. It was really interesting. So I'm wondering to what extent the intensity of interbreeding plays into figuring out things like the timeline and things like that. So if there was really frequent interbreeding versus it being a really rare event, how does that come into play when you're trying to use the level of introgression to figure out the timing of events?
Yeah, so I guess a question is this the case that this was an interbreeding event where there was a single interbreeding event involving a few Neanderthals mixing with 100 humans, or whether this was a prolonged event that happened over thousands of years? Right. So some of these results are they are robust to those types of assumptions. So things that we can say about how much ancestry we've inherited, or when this interbreeding event happened, they tend to give us accurate results, no matter whether this was a pulse of interbreeding over a short span of time versus a prolonged interbreeding event. So you can ask, if it's a prolonged interbreeding event that spanned, say, thousands of years, what date are we actually estimating? Typically, for these types of methods, they are most sensitive to the last date. So if it happened over tens of thousands of years and then it stopped 40,000 years back, typically these methods would pick out 40,000 as the date of last interbreeding between humans and Neanderthals. There's another interesting question, which is, can we actually say something more about the dynamics of interbreeding using these kinds of techniques? Was it the case that you had a large population of humans interbreeding with, say, hundreds of Neanderthals, or was it 100 humans interbreeding with a single Neanderthal? There's been some work, but not enough. It turns out that there is information in these kinds of maps that are informative of the dynamics of interbreeding. Roughly what might happen is, if it was a small founder population of humans encountering a small population of Neanderthals, essentially you will see like a lot of shared variation in the Neanderthal ancestry across humans today. Whereas if it's a large human population meeting lots of Neanderthals, there's going to be differences in the Neanderthal ancestry that different people carry. My guess is it's the former, so based on some preliminary analyses, but it's not something that we have actually, I mean, me or others have actually looked into carefully, but there is information available that should allow us to understand these kinds of questions.
You want to go first? I was curious about, I think it was your later slides where you said you were using the modern day genomes to infer amounts of archaic introgression in the different individuals. And I wondered about, yeah, exactly. I wondered about going forward, how you were applying this to populations that had larger degrees of admixture, particularly those in the Americas, compared specifically to Africans.
Yes, right. So these types of methods, they are sensitive to being able to figure out which is the majority ancestry and which is the minority ancestry. So often when we apply these methods, you get an ancestry, and then you have to interpret them. So you need to figure out if the method says there's 90% ancestry X and 10% ancestry Y, is the minority ancestry the archaic or the other way around? The reason we are able to do that is because we often have fairly good reference genomes for modern humans, so that we are then able to compare modern human genomes to these inferred ancestral genomes and say, okay, is the 90% archaic versus modern human? So that's where, so these types of analyses turn out to be important in interpreting this, where by doing these kinds of comparisons, we are able to say, is the minority ancestry archaic or the other way around? Yeah, so those kinds of, that's an important part of the interpretation of the predictions that come out of these types of methods.
Hi, thanks. This is really, really interesting. Thanks so much for the talk. I have kind of two questions, but it can like almost be grouped into one. So I was wondering if you were able to, it might not be possible because of sample sizing issues, but look at the extent of purifying selection and how that changed in different populations depending on like kind of like environmental constraints and pressures. Because I think with the UK Biobank, was it mostly Europeans that you did?
Yeah,
Okay.
Europeans, specifically white British. So
Oh, okay.
yeah.
And do you have enough to look at how the effects of the Neanderthal introgressed variants had, like, whether their impact on the complex phenotypes changed in like Asian populations? Because I think they have a sizable subset there too, and maybe the underlying genetic architecture would have.
Right. So that's an understudied question and really interesting. So if you go back to these maps, so the red is Europeans, the maps in Europeans, the green are the maps in East Asians. What comes out of this is there is some shared component. So you'll find some of these peaks lining up, but there are actually many places where the peaks don't line up. Part of this could be independent drift in Europeans and East Asians, and part of this could reflect selection pressures that are different in these populations. So yes, so I think it would be really interesting to be able to compare these kinds of mutations, not just in terms of differences in purifying selection, but also in terms of specific traits that they have impacted. So we know a little bit about it, but not quite enough. We do know that there's actually some small evidence of differences in the proportion of Neanderthal ancestries across different populations. So East Asians tend to seem to have a little higher Neanderthal ancestry than Europeans. That was previously interpreted as some type of purifying selection, although now it seems to be more of a statistical artifact of the way these ancestries have been identified, because they were identified in reference to African populations, and now Africans seem to have some European ancestry, which makes it challenging to identify cross-population differences. The idea of doing something like what we did in the UK Biobank with East Asians, I think it's really interesting. We only analyzed the white British individuals in the UK Biobank. It turns out the UK Biobank also has other ancestries, East Asian ancestries, South Asian ancestries, though not at the same sample sizes available for the white British. But I still think that's an open question, and the data is there to start answering that using similar types of approaches. And of course, there are biobanks that people are now beginning to build in other populations. So there are efforts going on to build biobanks in Japan and China. So I think that kind of data is imminent. But yeah, so it's a question that is waiting to be answered in some sense. And I guess the other thing which I should say in terms of time, the other important aspect here is I mentioned that we are now also getting access to ancient modern genomes. So these are individuals who lived thousands of years back, tens of thousands of years back. And now we can begin to ask questions about what the Neanderthal ancestry was in these ancient modern genomes. So now you get access to evolution happening at different time scales. And now you might be able to understand how purifying selection worked at different points of time. I think there's a question on the on Zoom. Yeah.
Oh, hold on. Wait. I think we had to unmute you here. Dan, can you try to talk again now? Nope.
The volume's the issue.
Oh, sorry. Thank you.
How about now?
Yes, now you're on. Thank you.
Okay, all right, thank you. So thank you for the talk, and forgive me if your questions either naive or confused. This is way outside my wheelhouse, but it seems germane to both your most recent remark and many of your remarks in the discussion thus far, who the Europeans are. So as a consumer of some of this literature, my understanding is that Europe was initially populated by anatomically modern humans, hunter-gatherers, and then there was a subsequent wave of influx of agriculturalists tens of thousands of years later who largely displaced that original hunter-gatherer population, which was, there are remnants of in northern Scandinavia, for example, right, sort of refuges where agriculture wasn't particularly viable. So it would seem that it was that population that contributed, that is, an ancestral human hunter-gatherer population in Europe that contributed to the flow towards East Asia, right? Because those anatomically modern humans would have moved eastward, right? And similarly, if you're trying to calculate the strength of the purifying selection, looking at that population, it seems to me, would be a stronger place to look, because the original admixture would have been greater there rather than filtering through many subsequent mating events. Does that make any sense?
Yes. So I should.
Yeah, I'm just going to mute this again. Sorry, Dan, before.
Right. So the question is present-day Europeans, and for that matter, present-day East Asian populations today themselves are the result of migration and admixture events that happened in the last tens of thousands of years. And so the question is whether the ancestral populations that contributed to these admixture events might have different proportions of Neanderthal ancestry, and the signals that we are detecting might differ across these different ancestral populations. Yeah, so that is definitely a possibility, and this is where the point that I was making about these ancestral modern genomes, ancestral modern human genomes, becomes important to consider these kinds of questions. So now that we are getting genome sequences from ancestral farmers, these ancestral hunter-gatherer populations, we can now begin to go back and try to understand what Neanderthal ancestry looked like in these populations and study the dynamics of Neanderthal ancestries in these ancestral populations. So continuing along those lines, for example, when we, so this is work that has looked at these, some of the earliest ancient genomes present in Europe, and typically these ancient genomes seem to be almost equally closely related to ancestral hunter-gatherers and ancestral farmers, and they seem to have about the same proportion of Neanderthal ancestry that was present as is present in present-day humans. So that seems to suggest that the Neanderthal ancestry happened prior to the split of the out of Africa population into all of these different populations, and to that extent it is a shared event. But of course, in terms of the dynamics, in terms of selection, it could have had different amounts of selection happening in these different populations. So yes, I think there's a lot of interesting complexity and nuance here, and that's something we are beginning to understand as we get access to these very diverse data sets across different points in time and space.
Thanks. Sorry if this is not also a very informed question, but I was just thinking like taking a big picture look. Like it seems like you talk a lot about events, like it's this binary, and I know for modeling purposes you always have to simplify things, right? However, we know like a lot of migration events are like repeated and can last really long periods of time, and then it seems like also you have much trouble modeling these introgression events as not just one-time things, but things that flows back and forth over tens of thousands of years, maybe. I was struck by this when you were doing your Deep Africa model, which seemed like a lot more complicated than the first model and seemed to fit the data a lot better. So I guess this is somewhat like more philosophical question, but what do you think about like the future directions of modeling in this like space R that can help account for like long periods of integration or long periods of mixing that are not like so starkly binary, but allow for like clouds of variation in populations?
Yeah, so I think that also ties into the question about what happens if these introgression events, which likely are not just a single pulse, they could have happened over an extended period of time, and they could have had migrants being exchanged between populations in both directions. So I think generally when we started out thinking about these kinds of questions, the first thing was the reality is likely to be complicated, which is what we are discovering. So the question is, are there simple models that we can build that can be falsified? So for example, you build a model where populations just split and nobody mixes. Here's a model, and is there any support for such a model from the data? So that turns out to be a convincing no. So now we ask, okay, can we now complicate these models? Can we now start introducing these introgression events and see which ones are supported by the data and which ones are not? And so that's been our general approach, where it is indeed the case that this is reality is complex, but the question is, can we now build increasingly complex models, starting with simpler ones, and see which ones are supported by the data and which ones are not? And yes, so I guess that's been the trajectory, and I think that's I think what we need to now wrap our heads around is how to think carefully about these increasingly complex models. How do we actually test them in a rigorous way? How do we set them up and see whether they can be supported by the data in specific contexts? So I think that's kind of the evolution of the way we've been thinking about these problems. But yes, as we look at the data, we're going to find more and more complexities, and we have to figure out how do we take our model and get it to match the complexity.
Brian, sorry.
So when you were discussing your Neanderthal results, one of the first things you mentioned was the low effective population size that's been estimated for Neanderthals. I was just wondering how might that, let's say if they did have a low effective population size, that could possibly lead to things like population bottlenecks, reducing diversity across all Neanderthal populations through time, let's say. How might you incorporate that into the later analyses that you did where, for example, you looked at a mismatch between Neanderthal and modern human loci, and then compare that to the chimpanzee as an outgroup? Is there the possibility, for example, that amongst all Neanderthal populations, there might have been greater genetic diversity at some point that is not visible to us today because of our limited sample of Neanderthal DNA? So that's a two-part question, because I actually don't know how many different samples of Neanderthal DNA we have, how deeply it goes in time, and how confident we should be in saying we know really that how diverse Neanderthals were, or if there is a sampling issue there.
Yes. Yeah. So that is always, so there are, I guess, a few parts to that question. So let's first start with what we know about Neanderthal genomes today in terms of how many genomes we have and where they're sampled from. So when we talk about Neanderthal genomes, again, there is what we can think of as high quality Neanderthal genomes, so high coverage Neanderthal genomes, where we are fairly confident about all of these, the bases that are present, and then we have many more lower quality Neanderthal genomes. So today we have 4 high coverage, high quality Neanderthal genomes, and the important thing about that is these have a fairly good span in terms of geography and time. So for example, the most recent Neanderthal genome, high coverage Neanderthal genome, is the one from Croatia, so that's about a little more than 30,000 years old. One of the early high coverage Neanderthal genomes that was from something called the Altai Neanderthal, that's about 100,000 years old. And now we have genomes from Western Europe, Central Europe, and Siberia across these time spans. So this is about the high coverage ones. We have many more lower coverage Neanderthal genomes. So now we are getting a better picture of diversity that exists within Neanderthals. And it generally seems to be the case that these different Neanderthal genomes are all sharing these similar characteristics of low diversity and being relatively similar to each other. So a lot of these analyses, and I didn't get into the technical details, we often repeat the analyses with different Neanderthal genomes to see how consistent they would be and how dependent they would be on the choice of which Neanderthal genome we've chosen to look at. For example, a lot of my analyses that I presented early on, we looked at the Altai Neanderthal, which is from the Denisova cave, confusingly the same cave where the Denisovan genome was discovered. And we've also then repeated it with the Vindia Neanderthal, the one from the cave in Croatia. So yes, again, it's still possible that we have newer Neanderthals which fall outside the range of genetic variation that we have seen, but I think the picture is relatively consistent in terms of them having low effective population sizes and fairly closely related to each other in spite of this spread in geography and sampling time. The other thing which also is good to think, good to point out in these types of analyses is when we think about, so there are 2 types of analyses that I've presented here. So one is analyses that are genome wide. So when we ask how much of the genome comes from Neanderthals or when this introgression event happened, they are looking across all of the genomes that we have. And this is it's counterintuitive. So the often a question that comes up is how are we able to make confident inferences when we have one Neanderthal genome sequence to begin with? And the reason is, although it's one genome, these kinds of inferences are looking at all the possible ancestors that this one individual had. And this is because of this recombination that we talked about. So you have one genome, but if you look at the first position versus the millionth position, because of recombination, they have very different ancestral histories. So these kinds of analyses are essentially using the fact that you're looking at one genome, but there are often thousands of ancestors of this one genome, and you're looking at all of them together to build confidence in these inferences. So that's also something which, it's counterintuitive, but it turns out to be powerful in being able to make these kinds of statements. The final thing is about how these low effective population sizes, how they affect some of these analyses. So essentially, and this is something also I should say is kind of a fortunate accident. So a lot of these analyses, again, the reason why we are able to make these analyses possible with single genomes is because of the fact that they have low effective population sizes. In other words, if you imagine that the Neanderthals were extremely bottlenecked, then one genome is fairly representative of the population, right? So that means we can now generalize our findings and say this is a finding that is generalizable across the population because of this low effective population size. On the other hand, if Neanderthals were extremely diverse, then it would actually be harder to do some of these analyses. So this is almost like an evolutionary accident, which has been helpful in these types of analyses. It may not generalize in other settings. When you look at other species, other populations, these kinds of methods may not be as effective in those settings.
There, we're out of time. Thank you so much.